Okay, today we're going to look at transcription and protein synthesis. So just as an overview, this is nothing new. We looked at this in your first year already. How are proteins made? So as you know, it's all done in the cell, and the information comes from our DNA. So we have our genetic information here. It varies depending on the type of organism, whether it's a prokaryotic or eukaryotic organism. So for prokaryotes, you know that everything is within the cytosol, but when it comes to the eukaryote, there is separation of the genetic information from the rest of the cell. And so the whole idea is the same. You have the genetic information. A copy of that information is made here through the creation of RNA molecule. So we say it is RNA synthesis, and the name given to this process is transcription. Now, after the creation of this RNA molecule, because it is taking the information to the cytosol, say for instance it's a eukaryotic organism, we call it the messenger RNA. Now, after entering into the cytosol, we have the ribosome, which is responsible for the creation of the polypeptide chains, as you can see in the illustration here. So the amino acids, they are brought to the complex by the transfer RNA molecule, and we'll look a little bit more at the transfer RNA molecule in a few. But as you know, the transfer RNA brings it to the complex, and after it brings it to the complex, the ribosome collects it, and it forms the polypeptide bonds, which creates this chain you are seeing here. Now, okay, let me go to the next. Sorry, yes. So before going into how proteins are made, once again, it is important to remember what nucleotides are. I touched on this a little bit in the last lecture when I was looking at the nucleoside triphosphates. And so, as you know, or as you should know by now, a nucleotide is that organic molecule that serves as the monomer or the subunits for nucleic acids. So nucleic acid is a biomolecule, and some examples would be like DNA and RNA. So let's look at the components of a nucleotide. So as I would have illustrated in the last class, you have the 5-carbon sugar, and attached to the 5-carbon sugar is the nitrogenous base. And together we say that these two are nucleosides. So these two can exist alone, and then sometimes you have them existing with that phosphate there. So when it has that phosphate there, that is when we say it is a nucleotide. So I went through this in the last lecture as well, just for a little, just once again to emphasize the structure of the nucleotide and the nucleoside. Now, the thing with nucleotides is that they serve as energy, right? I spoke about this already. So they carry packets of energy in the form of nucleoside triphosphates. And so it all depends on which base is attached here, and that's how it's given its name. So I went through that already. I wouldn't go much into that again. Now, let me just clear again so I can continue. Okay, good. Yes. Now moving to RNA and protein synthesis. So as I mentioned just now, both DNA and RNA are nucleic acids, and there are some things that sets them aside from each other. So these in the middle here are the features that I'm looking at. So firstly, when it comes to the sugars, DNA, they have deoxyribose, RNA is ribose. These are all things you should know by now, right? The nitrogenous bases, sorry. So when it comes to DNA, as you know, adenine, thymine, cytosine, guanosine, but the thymine is only found in the DNA and the uracil is only found in RNA. In terms of their strands, RNA is single-stranded, DNA is double-stranded, if you can remember from the very first illustration I had there as well. In terms of location, RNA, you can find them anywhere in the cell in numerous organelles. But when it comes to DNA, it's only in the nucleus, right? Okay, let's move to the next slide. So when it comes to the types of RNA now, I mentioned the messenger RNA, which is actually the copy that carries the genetic information for further processing. We also have the ribosomal RNA that makes up the ribosome, and we have the transfer RNA, all of which I mentioned earlier. The transfer RNA is the one that carries the amino acid to the ribosome. So let's go into the transcription process, which is the creation of the mRNA or the RNA synthesis process, right? So firstly, in order to get the information from the gene, in order to take it to the cytosol where the ribosome is going to create the protein, the messenger RNA needs to be produced, which is what I showed you earlier. So the messenger RNA, what it does is it's basically a complementary copy of one of the DNA strands. And this process is called transcription. So remember, in replication, both strands are being copied, but in transcription, only one of the strands are being used. It is important to note that the strand that is being used is called the template of sense. Now, when it comes to genes, there is a specific mRNA made under the control of the DNA, right? And for each of the genes, only the template of strand is actually transcribed. But the template of strand, sorry about that, may be different for different genes on the same DNA. So remember, when we have a genetic information, let me just try to illustrate this to ensure you are following. So when you have your DNA strand, on this DNA strand, you have numerous genes. From here to here might be a gene, from here to here might be a gene. They vary in length, right? And so you have variant genes, and for each of these genes, their template of sense differs. It could be on different strand because remember, this here is it's double stranded, right? So if for this gene, let me change the color. So if for this gene here, say this is the template of sense, then for this gene, it doesn't need to be this one. It might be this this next strand here that actually has the template of sense, right? So that is what it means by the strands may differ depending on different genes, or the strands that are being transcribed may be different for different genes. Now, as you know, a transcription, at the end of it, it just basically produces the mRNA. It also produces rRNAs, and these are, as I mentioned before, the major components of the ribosome. Moving on. Okay, so we're going into the steps in transcription, and the first step is the initiation, then we have the elongation, and finally the termination. So when it comes to the initiation, we have RNA polymerase. Make sure you note the difference in enzymes, right? So before I mentioned DNA polymerase, now it's RNA polymerase. So it all depends on what is being synthesized. If DNA is being synthesized as in replication, you have DNA polymerase doing this. If you're synthesizing an RNA, then you have RNA polymerase synthesizing that strand. So please do not confuse the enzymes with the process. DNA polymerase makes DNA, RNA polymerase makes RNA, right? It doesn't get simpler than that. So in order for the RNA polymerase, just like the DNA polymerase, in order for it to start working, it needs a recognition site, which is the primer. You know that we have an enzyme that prepares that for it. So similarly with the RNA polymerase, it requires that special sequence in order to start creating or synthesizing the RNA. And that sequence is called the promoter, right? So when they so now when each gene they have one promoter, and the promoter is actually what tells the polymerase where to start, which strand is the template of sense, all of these things, the promoter, the recognition sequence there, it will tell the polymerase all of these things, right? So the promoter, it has an initiation site, and this is a site where transcription will begin. And as you know by now, when it comes to polymerases, they move in that the 3' to 5' direction along the template strands. So if we look at the illustration here, let me just clear all previous drawings. Good. So if you look at the illustration here, we have the DNA. And so this particular gene here is being transcribed. The gene in this region here is being transcribed. And if you look, you can see we have the promoter in this region here. And as I mentioned, there is an initiation site, right? So it's just a recognition sequence you have at the beginning of the gene, and there is one for every gene. So on a genetic information, you can have multiple promoters, but each gene has one promoter. So this site here now, or the promoter site, so as you can see, the polymerase comes, it attaches at this initiation site here, and after it attaches here, the promoter is actually what controls the process after that. So it's going to tell the polymerase that, okay, this is where transcription needs to start. This is the strand. This is the strand that you need to transcribe. And remember, the promoter is going to be on the template of sense. So if you look, the promoter is going to be here and not here. If the promoter was on the top strand, then the top strand would have been used to create the messenger RNA. So it tells it which strand, and it also tells it if to go in this direction or if you're going in this direction, right? So based on the sequences that are here, the promoter can now know all of these things. Okay, let's move to the next slide. Okay. Yes. So once again, this is more information on the promoter. So when it comes to the efficiency of the promoted varies. So some of them they bind the polymers effectively, and some of them do not. And it all has to do with gene expression. I'm not going to go too much into gene expression with you guys, right? This course doesn't call for that, but if you go more into, if you move on to completed degree, that is where we're going to go more into the expression of genes and how it is controlled and how the promoter actually plays a part in this. And so, sometimes it all depends on the requirements for the cell, and that's how the efficiency of the promoter would vary. Sometimes you might need a protein might be in more demand at one time than it is in other time. It all depends, say for instance, if you're infected or not, right? If you have a virus or not, your body's trying to fight it off, it might need to produce more proteins as opposed to when you're not sick. So it has a lot to do with it. I'm not going to go into that and confuse you or whatever it is. So just remember the efficiency varies. It all depends on our bodily needs. Now, when it comes to prokaryotes, they have a single RNA polymerase. And that one polymerase, it creates everything. So it creates the messenger mRNA, creates the transfer RNA, and also the ribosomal RNA. But eukaryotes, as you know, we are a bit more complex, and so we have three different polymerases, RNA polymerases. The polymerase 2 is actually the one that is responsible for making the mRNA. Let me just clear that. Good. So we can say that transcription begins as soon as you have that polymerase binding to the promoter and it starts to unwind the DNA strand. Okay, so let's go to the elongation phase of transcription. Now the polymerase, it unwinds DNA at a fixed amount of bases at a time. So it depends on how many bases it can cover at a time. So usually there's 20 bases that it can cover at a time, and it reads a strand of the DNA in a 3' to 5' direction. So if I can show you this really quickly. So if you're looking here, you can see that this strand here is actually the template of sense, and so the polymerase attaches here, and it's reading from a 3 to 5 direction. That is what that means, right? Now the polymerase, in addition to that, it adds new nucleotides to the 3' end. So if you look here once again, you can see that this is the growing strand since it is reading, since the polymerase is reading the DNA in a 3 to 5 direction, then it will synthesize a strand in the opposite, because remember, it will be anti-parallel. So it is being synthesized in the 5 to 3, and as you can see, nucleotides are being added at the 3' end. Okay, let's go forward. So once the polymerase had processed a section of the DNA, it is very different from replication. As soon as a part of it is processed, it rewinds the strand, right? The polymerase is not as complex, the RNA polymerase is not as complex as the DNA polymerase. It does not inspect and correct, right? And so sometimes transcription, they occur at the rate of one mistake for every 10 base. So as soon as this part is finished transcribing, it moves to another 20 bases, and this part here rewinds. So it doesn't completely splits the DNA strand. And as I said before, nucleoside triphosphates are actually what are used to create, the new mRNA strand. Now the termination. So termination is specified by certain base sequences. Now let me just move to the illustration. Good. So you have the illustration here in order to terminate it. So where you see this red line here, this right here, so there is a particular sequence that is there, as you know by now, or you should know by now. Those are the stop codons. We will look at codons today. So when it comes to some genes, the newly formed transcript, it just falls away from the template when it reaches that stop codon. It just falls off, right? But for others, they require helper protein to pull the transcript from the complex. When it comes to prokaryotes, I would have mentioned this before last year, right? When you were in your first year. So I would have mentioned that for prokaryotes, the translation begins at the 5' end before transcription is complete. So if you look here, I think I should go to the illustration. Yes, right there. So as soon as this 5' end here, as soon as this end here for prokaryotes, as soon as this drops out of this complex here, translation begins, right? It's because remember, there's no separation of the genetic information in nucleus and so on. And so all of these, basically all of these molecules that are required to carry out these processes, they're all floating around in the cytosol. And so because of this, we can have translation beginning before transcription would have been completed. So for eukaryotes, because we have that separation there, we need to, the mRNA is made, and after the mRNA is made, okay, okay, Ronnie, I'm so sorry. I had no idea I was breaking up, but for the parts that you missed, you will have the lecture. So I'll pause the video right after the class so you can listen back and you can catch up back on the parts you would have missed. Right, so I was saying, I'm losing my train. Yes, I was talking about the eukaryotes. So they are separated. The genetic information is separated from the cytosol. And so there is the creation of the mRNA. And after the mRNA is created, it travels from the nucleus into the cytosol. We learned in the last lecture that for eukaryotes, there is the pre-mRNA, and the pre-mRNA is the one that has both the introns and the exons. And so before it is actually used, splicing needs to take place to remove all of the introns. Yes, so as I mentioned before, when it reaches the termination site here where we have the stop codons, the transcript is then set free from the entire complex, right? Moving on to genetic codes now. So the information that the ribosome needs in order to carry out protein synthesis, it lies in genetic code. So genetic code relates the genes to the mRNA and mRNA to the amino acids that will obviously make up proteins. Now, when it comes to codes, we basically say that they're codons. So it's 3 nucleotides, and we call these nucleotides codons. And each of them are found along the chain of an mRNA, and they specify a particular amino acid. Now, the codon on the mRNA is obviously complementary to the codon on the DNA from which it is transcribed, and I'll give you an example of this in a few. But before I change this, that is important to understand that codons, they're sequential, they do not overlap. There are 64 of them, but there's only 20 amino acids. So some of these amino acids, they have more than one codon. Some codons do not code for amino acids, but they do have special meanings. So I'm saying there's 64 codons that basically codes for protein. There are more than 64 codons, but only these actually are used in protein synthesis or codes for protein. So for instance, in the kidneys and stuff, we have varying amino acids there being made up of different codons, but those are not used in the synthesis of protein. So continuing with the genetic code, AUG is one of the simplest codons to remember, and it codes for methionine. Methionine is the start codon, and it's actually the codon that initiates translation. I spoke about the stop codons earlier, and these are UAA, UAG, and UGA. So whenever a complex, whenever whatever enzyme is doing the transcribing or the translating, as long as they meet one of these codons, as long as they meet one of these, termination will take place. So genetic codes are also said to be degenerate. And degenerate just simply means that an amino acid may be represented by more than one codon. I said this before. Now, when it comes to degeneracy, it doesn't only apply to the codes, but also the amino acids, because some amino acids have one codon, for example, methionine, while some may have numerous codons, for example, leucine. Okay, let me just clear the drawing. Okay, good. Yes, so as I would have said, degenerate just means that there is more than one way to specify an amino acid. Right now, when it comes to mRNA, we have the, if you look here, you can see that we have all the codons here, including the start codon, the stop codon, and so on. But before we go to this, let me first, okay, it's freezing, sorry about that. Yes. Now, it is important for you to understand before going into the table that these codons that are in the table here, they are complementary and anti-parallel to the codons on the DNA strand. And so, say this is the DNA strand, just as an example, and they come in pairs of 3. And so let's say we have A, A, A on the DNA. So this is our DNA. Yeah, so this is our DNA. Let's say we have A on the DNA. Then after the RNA polymerase attaches, it will basically create a strand that is anti-parallel to this master strand here. And so on your RNA, we will have U, 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 right? So AAA, which codes for phenylalanine, will be UUU on the messenger RNA, right? There's some other examples, like, yeah, there is also the example of ACC on the DNA, which will be, this codes for tryptophan, it will be UGG on the mRNA. And so the base pairing rules comes in here that we learned in your first year. So if you can remember, can anybody tell me what the base pairing rule is? Well, oh gosh. Well, I just gave the first one is that A always forms the bond with U and G always goes with C, right? Okay,
Yes, miss.
okay, good. So let's move on. Okay, so moving to the table now. Now, if you look at this table, you can see we have the first letter, we have the first letter here, we have the second letter at the top, and we have the third letter here, right? So let's look at the first letter. Let's look at methionine. Now, methionine is right here. And methionine, as I mentioned before, it is the start codon. So methionine is, the codon for methionine is AUG. So if you look at the first letter is A, so you'll find it here. The second letter is, let me just move the strip at the top, yes. So the second letter is U. If you look at the second letter, you can see that U is here. And then we have the third letter, which would be G, and that is why you have it in the bottom here. Now let's look at another example. Let's look at proline, for instance. Now when it comes to proline, the first letter for proline is C, and so that is why it's in this row here. The second letter is C as well. So a lot of protein has a lot of codons, for to be specific. And so let's look at this one here. So the first letter is C, the second letter is C, and so the third letter would be C. Any questions as it relates to reading this table before I move on? Okay, well, let me do another example just to be certain. So if you look at serine over here, so serine, you have the first letter, it's A, so be in this row. So if you're following along, you can see that it's remembered. It's always good to remember the order of these bases. So UCAG is always the order. UCAG at the top, as you can see, and then to each column UCAG. So the first letter is A, the second letter is G, and the third letter is U. And so these would code for serine. Do you have any questions before I move from this table?
Please, ma'am, excuse me. So
Yes.
AGC is also serine.
Yes, correct. Remember I was saying that the amino acids are degenerate, meaning that sometimes we have more than one sets of codon coding for it. And so AGC is serine. AGC, sorry, AGU and AGC is both for serine. Take for instance threonine, there's four sets of codons that codes for it, which are these.
Okay.
Right? The only one that has one codon, as you can see, is the methionine, which
And
is
that's the start codon.
AG.
Yeah.
That's a start codon, and we also have three stop codons. Right? So those are the exceptions. All the rest, they have more than one codons coding for one amino acid. And so that is why sometimes it's so easy for diseases to, genetic diseases to be created, right? Because say for instance arginine is supposed to be there, right? And so instead of arginine, a serine, one of these letters would have been replaced. Okay, so you have, say for instance you have GCU, right? This is for arginine. But say for instance an error happened and it's replaced with C, then it would still be arginine, right? It would be an error that wouldn't cause like major deformities, but still that's an error right here, right? So say for instance, arginine now is replaced with serine. In this case, this would cause major deformities, right? And sometimes it all just comes down to three bases being replaced. And that is what will cause all of these genetic disease like sickle cell and all of these things that we're going to be looking at in this course. So let me don't get ahead of time, right? Any other questions before I move? Okay. All right, moving on to translation, which is also called protein synthesis. So as you know, translation, this occurs at the ribosome in the cytosol, and it begins only when the mRNA transcript is made available. And as I would have said before, this process can be controlled by the cell. It all depends on the demand for a particular protein. And that process is called gene expression, right? So now the translation of mRNA into proteins, it requires a molecule that would link the information in the mRNA with the specific amino acids in proteins, right? Okay, yes, I was saying. So translation, it requires a molecule that would cause the linking between the information in the mRNA with the specific amino acids in the protein. And that is done or it is performed by the transfer RNA, which is basically a very small molecule. It is made of only 75 to 80 nucleotides, right? So the transfer RNA, it undergoes its own processing as well, whereby you have the amino acids being attached to it. So something you should note is that when it comes to transfer RNA, it is specific to an amino acid as well. So say for instance, a transfer RNA that would carry serine will only recognize the codons for serine. If it's supposed to carry methionine, then only methionine amino acids it can carry. So if there's 20 amino acids, there are 20 transfer RNA types, right? And so it is very specific. And in order for it to be charged, we say that the transfer RNA is being charged. In order for it to be charged with the correct amino acid, it has to go through a processing itself, and we'll look at that in a few, right? So now when it comes to the amino acids now, they bind to the 3' end, which I'll show you in the illustration right after this. So it they bind to the 3' end, and there is also a midpoint. And the midpoint, it contains three bases, and we call those bases anticodons. And the anticodons is actually the part that comes in contact with the mRNA. So I'll show you in a few what I mean by this. Now, if you look at this illustration here, it is depicting three different models of the transfer RNA. The first one is the clover leaf model. So we call it a clover leaf. This one I'm talking about here. This is the clover leaf. So the clover leaf model is it's basically emphasizing the base pairing and so on. If you look at the bottom here, we have the anticodon region, and this is actually the part that comes in contact with the messenger RNA. So as soon as I skip to the next slide, I'll show you this once again so you can better understand what I'm saying. So we have the clover leaf, then secondly, we have the three-dimensional representation. This is emphasizing the internal regions. And the last one is showing the computer-generated space filling representation. And then we have the icon, lastly, we have the icon here that is used in this lecture. So there's numerous ways, as long as you see something like this, know that it is the transfer RNA. So yet again, we have the function of the transfer RNA. And the transfer RNA is the molecule, as I mentioned before, that would take the amino acid once it has been charged with the correct one. It takes it and it brings it in contact with the messenger RNA and the ribosome, right? And the ribosome is actually the one that does the linking. Let me just remove the drawing there. Okay, yes. So once the transfer RNA decodes this, in a sense, we can say decode the mRNA, and it delivers the correct amino acid. So if it was supposed to bring alanine, for instance, once it delivers that one, the ribosome actually forms the peptide bond between the amino acids, right? Now, before I go into the translation, let's just look at the charging of the transfer RNA, and it is done by activating enzymes. So the activating enzymes, these are actually a family of enzymes, and we call them the amino acyl transfer RNA synthesis. It's a family of enzymes because remember we're saying that there's a transfer RNA for every amino acid, and so this group of enzymes recognizes all of them and it does the charging for it, right? So for each also for each amino acid there is one of these enzymes, one of the amino acyl transfer RNA synthase. And so that is why I'm saying it's a family of activating enzymes. So let's go to the illustration. Now, if you look here, we have let me just get my pencil. Okay. So if you look here, we have the enzyme here, the amino acyl transfer RNA synthase, which is specific for a particular amino acid. In this case, these here, both the transfer RNA and this amino acyl transfer RNA synthase is specific for the amino acid alanine, right? If you look alanine right here. So on the enzyme, there is various sites. So as you know, there's active sites on enzymes, So that is why you're seeing this enzyme shaped like this in this illustration. So there's always the amino acid site right here. We have the ATP site and we have the transfer RNA site right here. Right? So the first thing that happens is, okay, so the first thing that happens is the amino acid and the ATP attaches to the enzyme. Only one of the phosphate is actually needed for this process, and so the other two is given off as pyrophosphate, or two phosphates, as you can see in the illustration here is given off. And so when pyrophosphate is given off, AMP remains, right? So together the three of them make up, sorry, the two and the one makes up the ATP. So after this happens, we say that the alanine is activated, it is ready to be charged onto the transfer RNA. So the transfer RNA that is specific for the alanine comes in, it attaches to the enzyme. When it attaches to the enzyme, because the enzyme is so specific to alanine, it ensures that this is the correct transfer RNA, and this is the correct transfer RNA. This is the correct amino acid. All right. After it ensures that the phosphate is given off, and a bond is formed between the two, and then after that happens, it is released and it can now go to the mRNA where it gives up this alanine molecule. After giving up this alanine molecule, it then re-enters this process all over again. And so this happens, or this takes place for every single amino acid. And these transfer RNA, they're reused for multiple reactions, right? So it doesn't get degenerated after the first use. As you can see, it is our body recycles it. Do you have any questions before I move from this slide? Any questions?
No, miss.
Yeah. Okay. So this just explains what the illustration above is showing, the one we went through just now. Now moving on, the ribosome. So I think when we're done with the ribosome, I will end class for today because it's after 3 already and I don't want to keep you back too late, right? And I have another class after this. So we're going to look at the ribosome, and when we come the next day, we'll go into translation. When we come on Wednesday, I believe. Yes, Wednesday. So let's look at the ribosome really quickly. So I mentioned already that the ribosome is actually the molecule that does the translating and forms the peptide bonds between the protein. It has a large subunit and a small subunit. Now, if you look at the illustration here, you can see that the ribosome is actually irregular in shape, and it is made up, as I said just now, of two subunits. There is the large one at the bottom and the small one on the top. Now, if you look here also, you can see that the ribosome, on the ribosome there is an E site, a P site, and an A site, right? And these are all sites for transfer RNA binding. So we have the A site, which is the accepting site. We have the P site where the bonds are being formed, and we have the elimination site, which is the E site. So remember, when a transfer RNA enters, it all starts with AUG. So at this point, the ribosome will be covering, the A site will be here. Now, after the ribosome moves, then the A site will end up sorry, the AUG will end up in the P site if it moves 3 bases. And then when it moves 3 bases again, it will end up in the E site. So the ribosome, it keeps moving. Let me just draw. It keeps moving in this direction. So at one point, this would be the A site. Then at another point, it will become the P site. Then at another point, another 3 bases, it will become the E site. So as the ribosome moves, okay, so as it moves these 3 more bases, eventually it's going to leave the AUG, right? So when does it, okay, so how does this happen? So when the AUG, for instance, is in the A site, what happens is you have the transfer RNA bringing the particular amino acid methionine into this site. After that happens, the ribosome moves to another 3 bases, and this will end up in the P site. So after this ends up in the P site, remember the ribosome would have moved to another 3 bases, and so these will be in the A site, and so another transfer RNA will come in, bringing in this particular um, amino acid here. So a bond now will be formed between those two, and that is how the process continues. Of course, we have numerous illustrations to show this, and we'll go into that when we come back, when I meet you guys again on Wednesday. So let me just end the class here for today.